Chapter 22, it's about the respiratory system. So the respiratory system consists of a series of tubes that helps to deliver air into the lungs so we can have gas exchange and we can gain oxygen and get rid of carbon dioxide. In order to transport these gases into our tissue, the oxygen and the CO2 into the lungs, we have to work together with the cardiovascular system. So respiratory system and cardiovascular system will form the cardiopulmonary system, and they help to deliver oxygen into our tissues and remove the CO2. The respiratory system can also help to regulate the amount of hydrogen ions that we have in our body, or the acid-base balance, and in order to do so, work together with the urinary system. Now, transport of gases, it happens through the blood. And we have oxygen that it is transported 98.5% bound to hemoglobin. And 1.5% of the oxygen will be dissolved in the plasma, and it will form something that we call the partial pressure of oxygen, or PO2. Carbon dioxide also will be transported, and 90% of the carbon dioxide will be hydrated, forming carbonic acid, and later on this carbonic acid will be dissociated into bicarbonate ions, and 5% of the carbon dioxide will be bound to proteins, including the hemoglobin, and 5% of the carbon dioxide will be dissolved in plasma, forming a partial pressure that we call PCO2. About the oxygen, it is very important for our cells to form ATP, and oxygen will be found in about 20 milliliters per deciliter of blood. Hemoglobin will help to carry these oxygen molecules when oxygen binds to an iron atom in the middle of the heme group. And one oxygen molecule can carry up to 4 oxygen molecules, and when it does so, it forms 100% saturation. And 50% saturation will be when it's carrying half of these molecules, or 2 molecules of oxygen. When oxygen is bound to hemoglobin, it will form a structure that we call oxyhemoglobin, or HbO2, and when oxygen is released from hemoglobin, it will form deoxyhemoglobin, or HHB. Carbon dioxide can be transported as carbonic acid, as we mentioned before, as carbamino compounds, and dissolved in plasma. 90% of the CO2 will be forming carbonic acid, and this is how you form it. So you have here 1 molecule of CO2 in this formula, then you combine it with 1 molecule of water, and then you will have H2CO3, or carbonic acid. And then later on, this carbonic acid is going to be dissociated into bicarbonate ions, which is HCO3, and hydrogen ions. Now, when the CO2 binds to the hemoglobin, it will form this compound HbCO2 that we call carbaminohemoglobin. Now we will have gas exchange within the tissues, and this is called systemic gas exchange. In this case, the purpose of having this systemic gas exchange is to unload the oxygen that the hemoglobin was carrying so that the cells in our tissues can use this oxygen. And then when the cells are generating energy, are generating ATP, they are also generating a byproduct that it is called CO2 or carbon dioxide. This carbon dioxide is toxic to our cells, so we need to get rid of it. And then the hemoglobin exchanges oxygen for CO2. So when it unloads the oxygen into the cells, it will be free to pick up CO2. And this happens at the capillary beds of our tissues. Now, whenever you have an increased levels of CO2 in your blood, like 10%, you will increase your respiratory rate. This is one of the key points for this chapter. So remember, like 5% of the hemoglobin or the CO2 will be transported, but in the plasma. But whenever you have an increase of CO2 because possibly you are doing exercise and you're generating more CO2, then you will increase your respiratory rate. And the CO2 gets loaded into the red blood cells using this enzyme, carbonic anhydrase. So when the CO2 diffuses into the blood, part of it is going to go into the red blood cells, and in the red blood cells you will combine CO2 with water. You form carbonic acid through this enzyme that is called carbonic anhydrase, and this same enzyme, carbonic anhydrase, it will convert this carbonic acid, H2CO3, into bicarbonate ions and into hydrogen ions. And when this enzyme is doing this conversion, you will have simultaneously within the red blood cells something that we call the chloride shift, in which you exchange bicarbonate ions for chloride ions. And then hydrogen will bind to hemoglobin, the hydrogen ions that are coming from here. Now, in oxygen unloading, you will have the hydrogen that is binding to the hemoglobin, and this will decrease the chances of oxygen to bind. And it will make the hemoglobin much easier to release oxygen. So when the oxyhemoglobin will arrive the capillary beds in the tissues of our body, 90% will be saturated, and then as it's unloading the oxygen, it will leave 75% saturated. So we will unload like 22% of our oxygen. And then we will have afterwards something that is called the oxygen or venous reserve, in which the oxygen will remain in the blood after it passes through the capillary beds, but it will go then into the veins together with this oxygen, 75% of oxygen. Now here we have another point for this chapter, the key point. So whenever you have low levels of oxygen, when you have hypoxia or low levels of PO2, your pH level will drop. So you will have low levels of pH, and in turn, your hemoglobin molecules will open up and bind less strongly to the oxygen molecules. So this will increase the ability of the cells to pick up the oxygen. So it will increase internal respiration. So the hydrogen is going to be important then, because every time that you have high levels of hydrogen, you will displace the oxygen so that your cells can receive more oxygen. So this is the diagram to show you what happens in the systemic gas exchange. So in here we have the tissue cells, and then in here we have the capillaries with the plasma and with the red blood cells in here. So in the tissue, whenever it's active, you will release CO2 7%, that it will go as dissolved CO2 in the plasma, and 23% will also go into the plasma, but part of this will go and combine with plasma protein, forming carbamino compounds, and then also it will go into the erythrocytes, so to combine with hemoglobin to form carbaminohemoglobin, or HbCO2. And also 70% of the CO2 that it was generated will go into the erythrocytes. But instead of forming carbaminohemoglobin, as you saw here on the top, it will start combining with water through the action of CAH, or carbonic anhydrase, to form carbonic acid. And then later on, this carbonic acid is going to be converted into bicarbonate ions and into hydrogen ions. And then at the same time, you will have a chloride shift. So you will bring in chloride ions in exchange for bicarbonate ions. And this is going to be given by this protein in the membrane of the red blood cells, that it is an antiport. So you can extrude bicarbonate ions in exchange for chloride ions. Now these hydrogen ions will be coming here and combined with this oxyhemoglobin, and then you will get rid of oxygen and will leave this hemoglobin together with these hydrogen ions. And 98.5% of the oxygen will go and diffuse from this form into the tissues, and the 1.5% that it was dissolved as a gas will come and provide oxygen to the tissues too. So as the systemic gas exchange, what happens in our tissue cells? Now, in the alveoli, we have also gas exchange, but it's going to be the opposite. You will get rid of CO2 and you will load oxygen. So the reactions then occur as a reverse of the systemic gas exchange. So in the CO2 unloading, what is going to happen is that as the hemoglobin gets saturated with oxygen, the affinity for this hydrogen that it was previously bound will decrease. And this dissociation of hydrogen ions from hemoglobin will help to form this bicarbonate, as you can see here. And then you will form the carbonic acid, and this carbonic acid is going to be dissociated into water and CO2. And then you will have a reverse chloride shift, in which the HCO3 will come into the red blood cells, and then you get rid of chloride ions. So afterwards, then you can exhale the CO2. And this is another key point for this chapter. So the carbonic anhydrase, it's an enzyme that it is found in the red blood cells that can increase or decrease bicarbonate ions. So this is what happens in gas exchange in the alveoli. So here we have a little chamber in the lungs that is called the alveoli. You will see it later on in this, how they look in this chapter. So the alveoli is filled with air. So this is a chamber that is filled with air. And then in here you have the capillaries of the tissues of the lungs. And then here we have plasma, of course, surrounding this erythrocyte. So the capillary blood will have dissolved CO2. And this dissolved CO2 will diffuse into the alveoli because you will have more CO2 here than here. So from more to less will move the CO2. Now, the carbaminohemoglobin that you have here will unload this CO2, and then this CO2 will go into the alveoli. Also, the plasma proteins that might be bound with CO2, the carbamino compounds, will get rid of this CO2. So around 23% of this CO2 was bound into hemoglobin or into plasma proteins. And then this chloride shift that happened here, the reverse chloride shift, in which you bring HCO3 or bicarbonate ions in exchange for chloride, is going to be important to bring these bicarbonate ions that are going to combine with this hydrogen that are coming from this oxyhemoglobin, and then you will form again this carbonic acid. So this bicarbonate that you brought from the plasma, you will combine it with these hydrogen ions, you will form again this carbonic acid, and through this enzyme, the carbonic anhydrase, you can break down this carbonic acid into water and CO2, and then 70% of the CO2 that you have from here, it will be going into the alveoli, and also the water at some point as vapor. If you put your hands on the front of your mouth and you exhale, you are exhaling CO2, but if you feel it, you can feel some vapor, some humidity in your palm, and it's because you're getting rid of this water that you form. Now, since you will have here this hemoglobin that it was bound to hydrogen ions, and you have here within the alveoli a lot of oxygen, you have more oxygen in here than in the plasma or in the red blood cells, the oxygen will diffuse from more to less. So from the alveoli into the plasma and then into the erythrocytes. So 98.5% will go combined with this hemoglobin, and then you will form oxyhemoglobin, and then you displace this hydrogen ion that it was found here before. And 1.5% of the oxygen, since you have saturated all your hemoglobin molecules, will be dissolved as oxygen, and it will form the partial pressure of oxygen. So several things can affect the association of hemoglobin to oxygen, like temperature. So at a normal body temperature in this graph, you can see that you have, as it's depicted here on the y-axis, you have almost 100% saturation of hemoglobin with oxygen molecules. When you drop the body temperature, you reach almost 100% or a little bit over 100% of saturation of oxygen, or sorry, of hemoglobin with oxygen molecules. And when you increase the temperature of the body, you decrease the saturation of hemoglobin with oxygen, because as you increase the temperature in any proteins, you decrease the chances of these proteins having their structural normal form, in which in the case for hemoglobin, the globin molecules are close to each other and then they can bind the oxygen. But you separate them with temperature and then you cannot bind as many oxygen. Now, the partial pressure of oxygen also affects the formation of oxyhemoglobin. So in the systemic tissues, we have a low partial pressure of oxygen, as you can see here in the x-axis. So we have around 40% in the systemic tissues. So you will have a saturation of hemoglobin, like 78%. And then in the alveoli, where we have more partial pressure of oxygen, like 100%, you will reach 100% of saturation of hemoglobin with oxygen molecules, or 20 milliliters of oxygen per deciliter of blood. The concentration of hydrogen ions, or the pH, also have an effect on the saturation of hemoglobin with oxygen. So our normal pH is between 7.35 and 7.45. So in here they're showing you that at 7.40 of pH you will have 78% saturation of oxygen, or of hemoglobin with oxygen. And when you increase the pH, when you have something that we call alkalosis, you have increased saturation of hemoglobin, and it's going to be the opposite. When you have low levels of pH, when you have acidosis, something that we call acidosis, you will have low levels of saturation of hemoglobin. So blood gases have a relationship with the rate and the rhythm of respiration. So this is just to show you the levels of CO2, the partial pressure of CO2 and oxygen that we have at a pH between 7.35 and 7.45, the normal levels of pH. So at normal levels of pH, we have 40 millimeters of mercury of partial pressure of CO2 and 95% of 95 millimeters of mercury of partial pressure of oxygen, or PO2. So whenever you have an increase in the CO2, you will stimulate the respiratory rate. And this is because we have chemoreceptors in the cerebrospinal fluid and in the blood that helps to detect the levels of CO2. And these chemoreceptors will stimulate some respiratory centers that we have in the brainstem, in the medulla oblongata and the pons, so we can increase the respiratory rate, so we can get rid of CO2. So this is another key point then for this chapter. So the most potent stimulus for increasing the respiration, the respiratory rate, will be CO2. So we mentioned before that the respiratory system also helps to regulate the acid-base balance, or the pH. So within this system that works together with the urinary system and also with the brain, we have these central chemoreceptors that sends this signal through these nerves that innervates the lungs, so we can increase or decrease the respiratory rate accordingly. So whenever we have CO2 going into our brain, this CO2 will stimulate these central chemoreceptors, and then they will produce a 75% change in the respiratory rate, because you induce increased levels of hydrogen ions. So this will be detected because we have these chemoreceptors that detects not only the hydrogen but also the carbonic acid. And the hydrogen ions then will help to stimulate the peripheral chemoreceptors, and that will produce around 25% of the respiratory response to the pH changes, while 75% of these changes will be detected by central chemoreceptors that we have within the medulla oblongata. So whenever we have acidosis, which means having a pH level lower than 7.35, we will have hypercapnia, or high levels of CO2, which is greater than 43 millimeters of mercury. This will increase our respiratory rate. So we are accumulating CO2, so we will have to hyperventilate so we can get rid of this CO2. And then whenever we have alkalosis, which is having a pH level greater than 7.45, we don't have as much CO2, so we have hypocapnia. So the PCO2 will be less than 37 millimeters of mercury, and you will decrease your respiratory rate, so you will retain CO2, so you can return the pH to the normal levels, which will be between 7.35 and 7.45. Now, this acidosis and alkalosis, or increased hydrogen ions or decreased hydrogen ions, can be caused by problems in the respiratory system. And when you have this, you call this respiratory acidosis or respiratory alkalosis. And consequently, you will do the changes in your body to bring this acidosis or alkalosis to homeostasis. Now, hyperventilation will be the corrective homeostatic response to acidosis, as I told you before. So you will get rid of CO2. And then this will move your formula where you had a lot of carbonic acid. So you will convert this carbonic acid into water and CO2, so you can exhale the CO2. And this will reduce your hydrogen ions. And that will raise your pH, so you will return to normal. In hypoventilation, it is the opposite. So you will reduce your respiratory rate because you have alkalosis. In alkalosis, you don't have as much CO2 as you need to maintain the pH, so to maintain these levels of carbonic acid and hydrogen ions. So in this case, you would shift your reaction to the right. So you will hyperventilate, so you can increase CO2, so you can combine it with with water, so you can form carbonic acid, H2CO3, and this carbonic acid will be converted into bicarbonate and into hydrogen because you need hydrogen ions, so you will raise your hydrogen ion concentration and then you lower your pH to normal. Like if you had 7.5 of pH, you will bring it to 7.45 or so. Now, a case for acidosis is something as well that we call as ketoacidosis, in which someone might have ketone bodies in their blood as a result of degrading fat molecules through fatty acid oxidation. And in this case, these ketone bodies, which are found in excess, will stimulate as well the respiratory centers, and then you will have something that is called Kussmaul's respiration, in which you have hyperventilation to get rid of excess of CO2 and to compensate for the acidity of the ketone bodies. So CO2 then has indirect effects on respiration through increasing the pH levels in acidosis, as we said before, but will also produce direct effects, in which whenever you have increased levels of CO2 because you're starting to exercise, you will stimulate your peripheral chemoreceptors, and then you increase your ventilations. And this will be more important because this will act more quicker than the central chemoreceptors. So you don't have to wait for changes in pH to start breathing faster when you need it. And oxygen levels have very little effect on respiration. So someone can be put into a mask with 100% oxygen, and the person's respiratory rate is not going to increase at all. But if someone starts being in an area where there is a lot of carbon monoxide or CO2 levels, this person is going to start breathing faster because CO2 is more important to increase the respiratory rate than oxygen. Oxygen is good for our body, but CO2 is toxic. So that's why we need to breathe faster when we have high levels of CO2. Now, when someone has chronic hypoxemia or having a partial pressure of oxygen of less than 60 millimeters of mercury, that will stimulate the ventilation, but it is because the person will have high levels of CO2. Examples of cases of chronic hypoxemia is when someone smokes and damages lungs and causes emphysema, when someone has pneumonia, or when someone lives in high elevations. Okay, so now let's talk about the anatomy of the respiratory system. So, as we said before, the respiratory system consists of a series of tubes that help us to produce gas exchange and to transport the gases in our body. And the functions of the respiratory system, besides gas exchange, will be communication, because we have several organs that help us to produce speech or other vocalizations. And also, as we already described, the respiratory system also helps to control the acid-base balance. Another function of the respiratory system will be to help to synthesize a molecule that is called angiotensin II that helps to regulate the blood pressure, and also the respiratory system helps to create pressure gradients between the thorax and the abdomen so we can promote the flow of lymph in the blood, as we mentioned in the cardiovascular system chapter, that it helps to form this respiratory pump or thoracic pump. So the respiratory system has main organs like the nose, pharynx, larynx, trachea, bronchi, and lungs. And inside our lungs, we have these chambers filled with gases that we call the alveoli. And we have millions of alveoli within our lungs. And these alveoli, they have a very thin wall made out of simple squamous epithelium. And this is where actually respiratory gas exchange happens, or alveolar gas exchange. Now, within the respiratory system, physiologically speaking, we have two zones. One that it can conduct only the gases, but we don't have gas exchange. And this will be called the conducting zone or anatomical dead space. And this zone goes from the nose up to the major bronchioles. And then we have this second zone that is known as the respiratory zone, in which we have gas exchange. And this consists of the alveoli and other gas exchange regions. Any organ in the respiratory system that is located above the larynx is called the upper respiratory tract, and anything below the larynx will be called the lower respiratory tract. This is the overview of the structures of the respiratory system. So in here we have the upper respiratory tract anything above the larynx. So we will have the nostrils and also the nasopharynx, which is this, the oropharynx, which is found in here, and the laryngopharynx, which is found in here, and of course the larynx. Below the larynx, we have the structures of the lower respiratory system, which will be the trachea, the bronchi and its branches, and the lungs and the pleura, the membranes that protects the lungs. Let's talk about the first structure of the respiratory system, the nose. So functions of the nose will be to warm, clean, and humidify the air that we inhale, and also help us to detect odors and acts as a resonance chamber, so we can amplify our voice. The nose will start at the nostrils or nares, which is the anterior opening, and then ends in the posterior opening of the nose that we call the choanae. Now, the nose anteriorly, the facial part, it is made by bone and hyaline cartilage. At the root of our nose, we have the nasal bones, and then the tip of our nose will be made of hyaline cartilage. So this is the external structure of the nose. So here we have the root made out of these nasal bones together with the bridge of the nose. And then we have this anterior middle portion that is called the dorsum nasal. And internally to this nasal, we have the hyaline cartilage. And then here we have the opening nares or nostrils. And then we have the ala nasal, which is made by connective tissue and also by minor cartilages. So these are the minor cartilages that forms part of the ala together with the connective tissue, and in here you can see these nasal bones and this lateral cartilage. Internally, our nose is subdivided into the right and left nasal fossae. And the structure that divides the nose into this fossae will be the nasal septum, which has a bony part that it is made by the vomer inferiorly and superiorly by the ethmoid, and then we will have the nasal cartilage that helps also to divide the nose into right and left nasal fossae. The starting point of the internal portion of the nose is called the vestibule, the internal area. And within the vestibule, we have stratified squamous epithelium that it is filled with these vibrissae, which are little hairs that helps to block the entry into our nose by insects and other debris. Above the vestibule and laterally, we have the nasal conchae, which are folds of tissue that helps to bring more air into the roof of the nose, and they are projecting laterally on the nasal fossae. Now, beneath this conchae, which we have three, which will be the superior, middle, and inferior, we have openings that we call, or spaces that we call meatuses. And then within these meatuses, we have the drainage either from the paranasal sinuses or from the lacrimal duct. Now, another function of the turbinates or conchae will be to increase the air turbulence, so you can have more air going into the roof of the nose, but also to clean, warm, and moisturize the air that we inhale. Now, the entire nasal fossae, with the exception of the roof, will be covered by the olfactory epithelium, which is made out of pseudostratified columnar epithelium with globed cells that produces mucus. And within this pseudostratified columnar epithelium, in the roof of the nasal fossa, we will have specialized cells that act as sensory cells to detect odor molecules. Now, the nasal fossa also has this venous plexus that helps to humidify the air and also to help to warm the air every around 30 and 60 minutes. So this is the section of the respiratory mucosa to show you the type of epithelium that we have. This is pseudostratified columnar epithelium with cilia and gloved cells, which are these. The gloved cells, they secrete their mucus coat into the apical portion of this respiratory epithelium so we can protect the cilia. The cilia helps to sweep away the mucus and also to trap any debris. So this is a better magnification of the nasal area, so you can see the internal structures of the nasal fossa. So here we have the roof of the nose where we have this respiratory epithelium, and then we have the superior, middle, and inferior nasal conchae with the corresponding meaduses. So we have superior, middle, and inferior meaduses. Here in the superior and middle meaduses, we have the drainage of the paranasal sinuses, while in the inferior, we have the drainage of the lacrimal duct. And then here is the vestibule with the vibrissae, and then in here we have the posterior opening that is called the choanae. This is the lateral portion of the medial portion of the nasal septum. So you can see here that we have the septal cartilage, and then in here we have the ethmoid bone with its perpendicular plate, and then in here we have the vomer. The next portion of the respiratory system is the pharynx, which is made out of muscle. So it's a muscular funnel that measures around 5 inches in length. And it runs from the choana, the posterior nasal aperture, up to the larynx. Now the pharynx is subdivided into 3 major regions. The nasopharynx, the one that is posteriorly located to the nose, then the oropharynx, the one that is posteriorly located to the oral cavity, and then the laryngopharynx, that it is communicating the oropharynx to the laryngopharynx. One characteristic about the nasopharynx is that it is lined by respiratory mucosa, pseudostratified columnar epithelium. And in here, we only have the passage of air. Now, within the nasopharynx, we have pharyngeal tonsils, and we have an opening which is communicating the middle ear to the pharynx. These are the auditory tubes or Eustachian tubes. And then the nasopharynx, we have a 90-degree turn that helps to trap larger particles. The oropharynx and the laryngopharynx, they both pass food, liquid, and air, and they are covered by a stratified squamous epithelium. A stratified squamous epithelium is a stronger epithelium than the pseudostratified columnar epithelium, and since we have here chances of friction because we are passing food and liquids, we need to have this type of epithelium. Now within the laryngopharynx, we have also the starting point for the esophagus, which is part of the digestive system. The next portion of the respiratory system below the pharynx will be the larynx or voice box. The larynx has a length of 1.5 inches, is much shorter than the pharynx, and one of the functions of the larynx is to produce voice, but also to keep food and drinks away from the respiratory system. And in order for us to produce voice, we have within the larynx different type of cartilage. Now, for us to prevent having food and fluids going into the respiratory system, we have this cartilage that is called the epiglottis. So the epiglottis will guard the entry into the larynx. And when we are at rest, this epiglottis is almost found in a vertical position. But when we swallow or drink, we will have some muscles from the larynx that will pull the larynx upward, and then that will help together with the tongue to push the epiglottis down so you will close the larynx. And then inside the larynx, we will have some folds of epithelium that we call vestibular folds that helps also to keep away anything that might have gone into the larynx of food and drink away. So this is the different views of the larynx. So we have here the anterior view, this is the posterior view, and this is the mid-sagittal view. So the larynx will run from this region up to here. So in here you can see anteriorly two of the major cartilages of the larynx, which will be thyroid cartilage and the cricoid cartilage. These two are joined by this cricothyroid membrane, which is one of the membranes that they can open when someone has an upper respiratory obstruction, and then they can insert a tube so the person can breathe. Posteriorly and anteriorly, you can see the epiglottis, which is the first, or well, it's not the first, but this is the cartilage that we already talked about that protects the opening of the larynx. So this is the lateral view of the epiglottis. So when we are at rest, this is almost open vertically, this epiglottis, and then or position vertically, and then when we are swallowing food or drinks, we will close the larynx with the epiglottis. And then in here in the posterior view, you can see minor cartilages like these that are called the corniculate. Also you can see in the lateral view, and then you see the posterior part of the cricoid cartilage in here and of the thyroid cartilage. And in the mid-sagittal view, you can see also this cartilage that it is found in here that is called the cuneiform cartilage. And then the arytenoid cartilage, which is this one, and then the cricoid in here. And then below the larynx, we have the trachea. So in total within the larynx, we have 9 cartilages, and the first 3 are single and relatively large. So the epiglottis, the thyroid, and the cricoid cartilage. In males, once they reach puberty, the thyroid cartilage enlarges and forms what we call the Adam's apple. The 3 paired small cartilages of the larynx are the arytenoid, the corniculate, and the cuneiform. And they help to tense the muscles of our larynx so we can produce different pitches of voice. Now the larynx is suspended from the hyoid bone by the thyrohyoid ligament, by the cricothyroid, by the thyrohyoid ligament, and the cricotracheal ligament suspends this larynx into the trachea, and then the intrinsic ligaments will hold these laryngeal cartilages together. The sides of the larynx and posteriorly are covered by muscles, and these muscles are very strong, are called the intrinsic muscles, and they help to operate the vocal cords, which are also called as true because they will produce voice. Above the true vocal cords, we have the false vocal cords or vestibular folds that they don't produce voice. And the way that we produce voice is by passing air through these vocal cords, tensing these vocal cords, and vibrating these vocal cords. So one of the functions of these muscles of the larynx will be to pull on the corniculate and arytenoid cartilages so you can pivot them, and then you can abduct or adduct your vocal cords, and you can produce different pitches of voice. So the lower pitch sounds will be produced when your vocal cords are slack, and when you have them so tense, you can produce high pitch sound. There is a difference between the male and the female vocal cords. So in males, the vocal cords are usually longer and thicker, and they vibrate more slowly. So that's why they produce a lower pitch sound in comparison to the female voice. And then the loudness of our voice, it is determined by the force of air that passes between the vocal cords. Now there is a space between the vocal cords that is called the glottis. Don't confuse that with the epiglottis. And this glottis can open and close as you tense these vocal cords so that you can produce voice at different pitches, but also the voice will be modified by movements of the oral cavity, the tongue, the lips, and the pharynx. So this is a superior view of the larynx. This space in between these foldings, which are the vocal cords, is what we call the glottis. And in here we have the epiglottis, and then you can see the corniculate cartilage in here and here below the trachea. And these vocal cords are called true, while these ones, the vestibular folds, are called the false vocal cords because they don't produce voice, only these ones. So when you adduct your vocal cords, you close your glottis, and then you can produce a high-pitched sound. And when you abduct your vocal cords, when you separate and you open your glottis, and then you produce low-pitched sounds. Now the trachea. The trachea is also known as the windpipe, and it measures 4.5 inches in length. And the trachea, it is made by C-shaped rings. So this is another key point that you need to know. And we have between 16 to 20 C-shaped rings that are made out of hyaline cartilage. This hyaline cartilage will help to reinforce the trachea to prevent collapse of the trachea during inhalation. And these C-shaped rings are open posteriorly towards the esophagus and are separated from the esophagus by this muscle that we call the trachealis muscle. And the trachea is located anterior to the esophagus, and when the esophagus is passing food, it allows this trachealis muscle to expand anteriorly, preventing the food from getting stuck in the esophagus. And then the trachea can increase or decrease its diameter to adjust the airflow. So when you increase the diameter of the trachea, it will relax its walls so you can have more influx of air. The trachea inside is lined by pseudostratified columnar epithelium with cilia. And this cilia as well has, as any other cilia in our body, certain mobility that is in charge of this reflex that we call the mucociliary escalator reflex, that it is a mechanism for removal of debris. So whenever you have molecules that are trapped within these cilia, you start this mucociliary escalator reflex so you can get rid of this debris. So this mucociliator mucociliary escalator reflex is basically the movement of the cilia upward so you can drive the mucus towards the pharynx and then you can swallow the mucus. Now the middle layer of the trachea, it is made by this connective tissue that helps to hold the trachea into the surrounding tissue, and within this middle layer of the trachea we have also lymphatic nodules that help us to protect from infections, and then we have mucous and serous glands and the tracheal cartilage. So this is the histological view or microscopic view of the trachea. So this is the epithelium of the trachea, pseudostratified columnar with cilia and the globed cells that secretes the mucus into the surface of the epithelium so you can trap the debris. And then in here we have this connective tissue that it is made by lamina propria, submucosa, and perichondrium. And then in here we have cartilage. The other layer of the trachea is the adventitia, that it is made out of fibrous connective tissue that will blend into the organs of the mediastinum. Below the trachea, we have this splitting of the trachea into the right and left main bronchi. So the trachea will divide into the right and left main bronchi at the external angle in the second intercostal space. And then internally, this division can be seen as an internal medial ridge that we call the carina. And then from the trachea, you can drive the air into the right and left main bronchi. So this is the cartoon to show you the walls of the trachea, the composition of the walls of the trachea, and also the division of the trachea into the right and left main bronchi, which are these. So if you make a cross section of the trachea and you see it, you can see that you have here the epithelium, the lumen of the trachea, the trachealis muscle, and these C-shaped rings that are made out of hyaline cartilage. If you make a longitudinal section, you will be able to see the pseudostratified columnar epithelium with the globed cells, the cilia, and the mucus coat, and with this submucosa that is made by mucus glands and also by this cartilage, the hyaline cartilage. And then here is the division of the trachea at the carina into the right and left main bronchi or primary bronchi. Now these bronchi will bring the air into the right and the left lungs, and then the main bronchi will subdivide into the lower bronchi, and the lower bronchi will divide into the segmental bronchi, forming a bronchial tree. This is a scanning electron micrograph enhanced with color to show you the cilia of this tracheal epithelium. These cilia can be destroyed by microorganisms, or it can be damaged by smoking. So here you can see the main bronchi, how they are delivering air into the lungs. And now that we have here the lungs, so the right and the left lungs, they have different numbers of lobes. They are segmented. So this right lung has 1, 2, 3 lobes, while this left lung has only 2 lobes, 1 and 2. The name of the lobes in the left lung will be superior and inferior, while the ones in the right lung will be superior, middle, and inferior. And then this main bronchi again will be subdivided into the lower bronchi, superior, middle, and inferior in the case of the right, and superior and inferior in the case of the left. And then these lower bronchi will be subdivided into the segmental. So these lobes are subdivided of the lungs into the different segments. So the lungs, they have a conical shape with the base that faces the inferior portion of our thoracic cavity and an apex that faces superiorly. And then medially we have these indentations that we call the hilum. And in the hilum we have the entry of the pulmonary arteries and the exit of the pulmonary veins, and then the entry of the lobar bronchi. So this is the right lung. So you can see the apex, the base, and then the medial view or medial size, the mediastinal surface. And then these lobes of the right lung are separated by the horizontal fissure and the oblique fissure, while the lobes of the left lung are separated only by this oblique fissure. If you make a cross section of the thoracic cavity, you can see that the lungs are separated by the heart, the mediastinum. So characteristics about the lungs. So the base are broader and it has a concave portion that rests on the diaphragm, and the apex is pointed, so it forms a tip that projects above the clavicle. The lateral, anterior, and posterior surfaces are called the costal surfaces. They are pressed against the rib cage. And the mediastinal surface, which is the medial surface, faces toward the heart, and it will have the hilum that receives the main bronchi, the blood vessels, the lymphatics, and the nerves, and they will constitute what we call the root of the lung. So one difference between the right lung and the left lung is the right lung is shorter because the liver rises higher on the right side and pushes the lungs up, which makes them smaller. And then the left lung is tall and narrow because the heart tilts towards the left lung and occupies more space on this side of the mediastinum, and this heart will form an indentation in the left lung in the hilum that we call the cardiac impression. Now for the bronchial tree. So the bronchial tree is lined with ciliated pseudostratified columnar epithelium. So basically columnar epithelium. And as you move distally into the bronchial tree, you will have a change in epithelium. So the cells will become shorter and thinner, and eventually you will have a complete change of epithelium into cuboidal and then into simple squamous epithelium when you reach into the alveoli. Now the bronchi are surrounded by mucous glands and also by lymphoid tissue that we call mucosa associated lymphoid tissue or MALT. And this epithelium, it is subject to the entry of pathogens, so it has to have this protection with MALT. And all the divisions of the bronchial tree will have a large amount of elastic connective tissue that helps to recoil the lungs during exhalation so you can expel the air or exhale the air from the lungs. Now, outside of the mucosa, we have smooth muscle cells, and these smooth muscle cells are called the muscularis mucosae that contracts or relaxes to increase or decrease the lumen of the airways and regulates the airflow. Of course, when you have contraction of these muscularis mucosae, you will decrease the lumen and you decrease the airflow into the lungs. Now, together with the muscularis mucosae and the bronchi, you have the pulmonary artery branches that follows closely these bronchial trees so they can provide blood supply into the tissue that surrounds this bronchial tree. And then at the end of the bronchial tree, you will have the formation of these capillaries out of these pulmonary artery branches so you can have the gas exchange. And then the bronchial tree also will have this bronchial artery that will provide systemic blood. And this bronchial artery will be coming from the aorta. So the characteristics of the bronchial tree. So it's made out of a system of air tubes that are located within the lungs, and the main bronchus will subdivide into 65,000 terminal bronchioles. So we will start with the primary or main bronchi. This primary or main bronchi, it is supported by C-shaped hyaline cartilage rings, and one difference between the right and the left bronchi is that the right main bronchi, it is shorter and wider than the left bronchi. So because of this, and because of its position, which is more vertical than the left, most of the foreign objects that are aspirated or inhaled will tend to go into the right main bronchus because it's shorter, it points more vertically, and is wider. And then the left main bronchus is longer, is narrower, and it runs more horizontal than the right. Now the left and the right main bronchi will subdivide into the secondary bronchi that are supported by crescent-shaped cartilage plates, and we have 3 lower bronchi on the right lung and 2 on the left, and they have the same names as the lobes of the lungs. They have superior, middle, inferior names on the right and only superior and inferior on the left. The lower bronchi or secondary bronchi will subdivide into segmental or tertiary, and this tertiary we will have 10 on the right lung and 8 on the left, and they will supply a structure within the lungs that we call as the bronchopulmonary segment, which is an independent unit of the lung tissue. So we have 10 bronchopulmonary segments on the right and 8 bronchopulmonary segments on the left. These tertiary bronchi will subdivide into bronchioles. Bronchioles are tubes that have 1 millimeter or less of diameter, and they will provide air into pulmonary lobules within these bronchopulmonary segments. So pulmonary lobule will be a portion of the lung supplied by one bronchiole. And in here is where we have the exchange of epithelium. So instead of having pseudostratified columnar epithelium, the bronchioles have ciliated cuboidal epithelium. And in here we have well-developed layer of smooth muscle, and the bronchioles will divide into 50 to 80 terminal bronchioles. These terminal bronchioles will be the final branches of the conducting zone. So this is the end of the anatomical dead space. And these terminal bronchioles will be half of the diameter than the bronchioles. So we will have 0.5 millimeters. And in here we don't have mucus glands because the diameter is so small that can be plugged really easily with mucus. And in here we have cilia that will move any mucus that will be draining from the bronchioles into here, and then we will use the mucociliary escalator reflex to move this mucus from the terminal bronchioles up into the bronchioles. And each terminal bronchioles will give 2 or more smaller respiratory bronchioles. The respiratory bronchioles will have at the end these clusters of alveoli. And then in here is where we start having respiratory zone or gas exchange. So the conducting zone ends at the terminal bronchioles, but in the respiratory bronchioles we have respiration or gas exchange. And these respiratory bronchioles will divide into 2 to 10 alveolar ducts. And within these alveolar ducts is where we have the actual attachment of these clusters of alveoli that forms the alveolar sacs. So we have around 150 million of alveoli in each of our lungs, and it will provide 70 square meters of exchange surface for gases. Now, within the alveoli, we will have a different type of epithelium than the respiratory bronchioles. So we will have simple squamous epithelium. And in the alveoli, we will have different types of cells. The walls of the alveoli will be made out of 95% with type 1 alveolar cells. These cells are simple squamous because you need to have rapid gas exchange. So since these cells are very, very flat, the squamous epithelium, you can have rapid diffusion of gases between the capillaries and the alveoli. The other type of cells that we have within the alveoli will be type 2. These type 2 cells will produce a substance that prevents the alveoli from collapsing. This substance will be called pulmonary surfactant. This pulmonary surfactant will be made out of a mixture of phospholipids and proteins that decreases the surface tension of the alveoli, that if we didn't break this surface tension, the alveoli will collapse and then we will not breathe. This surfactant, it is produced in utero around week 23 or so, and then it will finalize by week 36 of pregnancy. And the babies, when they born premature, preterms, they have as more premature as they are, they have less chances of having surfactant. So you have to induce the production of surfactant so they don't have respiratory distress. Other cells that are found within the alveoli but are not part of the alveolar walls will be the macrophages that we call the dust cells, the alveolar macrophages. These are most numerous of all the cells in the lungs, and they are looking for anything that can be found in the alveoli, like debris or pathogens. We have so many dust cells that about 100 million dust cells die each day, and then as they die, they are riding up the mucociliary escalator reflex, and they are swallowed and digested with their load of debris. Okay, so these bronchioles subdivide into these terminal bronchioles, and in here is where you end with the conducting zone. So up to here you only conduct air, but you don't have gas exchange. And then in here, in this area, we have the respiratory bronchioles that are attached to these alveolar sacs. And if you make a section of these alveolar sacs, you can see these alveoli, which are little chambers that looks like honeycomb, that they are filled with air. And then these alveoli, or alveolar sacs, are surrounded by the capillary beds that are made by these branches of the pulmonary veins. And then in here, you have these capillary beds draining the oxygenated blood into the pulmonary veins, so that this oxygenated blood can return into the heart, so it can be delivered, the oxygenated blood, into our tissues. So this is where gas exchange happens. So the alveolus are surrounded by the capillaries supplied by the pulmonary artery, and then within the alveolar sacs, we have this respiratory membrane, which is made by the walls of the alveoli, and also by the walls of the capillaries, and by the basement membrane that they have in between, so that you can have this gas exchange. Now, one important function of the alveoli is that you don't have to have fluid in it, because fluid will not allow for gas exchange to happen. So you have to keep the alveoli dry by absorption of the excess of fluid by the blood capillaries. So the lungs will have an extensive lymphatic drainage that prevents these alveoli from being saturated with fluids, so you can have free gas exchange. And then within the alveoli, you have low capillary blood pressure that prevents the rupture of these uh, capillaries, preventing the filling of the alveoli with fluids. So this is then how this gas exchange happens within this respiratory membrane. So in here in the respiratory membrane, you can see the wall of the alveolus, which is simple squamous epithelium, the wall of the capillary, which is made out of simple squamous epithelium as well, and the basement membrane in between. So oxygen, it is loaded here. So within the alveoli, you have high amounts of oxygen. So you load these capillaries with oxygen, and then you get rid of the CO2, because the CO2 in the capillaries of the lungs, they are being filled with a lot of carbon dioxide from our tissues, and then you unload it in here. And everything happens by diffusion. So an area of high concentration of oxygen will diffuse the oxygen into an area of low concentration of oxygen, and an area of high concentration of CO2 will load the CO2 into an area of low concentration of CO2. And this is the histology of the lungs. So here you have the lumen of a segmental or tertiary bronchus. You can see here the smooth muscles surrounding these bronchi, the bronchial mucosa, and the bronchial cartilage. And then in here you can see the branches of the pulmonary artery and the alveoli. This is a huge magnification with an scanning electron micrograph showing you the bronchial walls and the alveoli. This is a light microscopy of the lung tissue showing you a conducting bronchiole, an alveolar duct in here. So this is the conducting bronchiole, the alveolar the alveolar duct in here, and then in here you have a branch of the pulmonary artery and the alveolar sacs in here surrounding this alveolar duct, and then the alveolus in here. All of these will be the alveoli. If you have a very huge magnification of the alveoli, you can see these alveolar walls, how thin they are. They are made of simple squamous epithelium, and then you can see some alveolar macrophages in here. And this is again another diagram showing you the structure of the respiratory membrane, made out of the wall of the alveoli, simple squamous epithelium, the walls of the capillaries, and the basement membrane in between. And then you have here the alveolar macrophages and gas exchange through diffusion. So CO2 is exhaled, oxygen is inhaled and brought into the capillaries. So this is the respiratory membrane: squamous alveolar cell, capillary endothelial cell, and shared basement membrane. It's very thin, so that's why you have rapid diffusion. Now let's talk about the covering of the lungs. So the lungs are covered by pleuras. So we have the visceral pleura and we have the parietal pleura. Visceral pleura is the inner membrane, is a serous membrane that covers directly the lungs, and the parietal pleura is outside the visceral pleura, and in between you have the pleural cavity, which is a potential space. This pleural cavity is filled with pleural fluid, and you prevent friction with this pleura as the lungs expand within the thoracic cavity. And also you create a pressure gradient, so you lower the pressure greater than the atmospheric pressure so you can inhale. And then this pleura also helps to prevent the the spread of infections from one organ in the mediastinum to others, or in the thoracic cavity to others. So similar as to the pericardial membranes, you have a double membrane that surrounds the lungs. In the case of the pericardial membranes, they surround the heart. So this is the visceral pleura in close contact with the lungs, and outside you have the pleural pleura, and in between you have the pleural cavity. Now for ventilation, which is the movement of air in and out of the lungs. So we have pulmonary ventilation that consists of inhalation and exhalation, and they form together the respiratory cycle. So we have within the respiratory cycle quiet respiration that happens when you are at rest, and usually one part of this quiet respiration, it is effortless. You still have to use your inhalation muscles to have quiet respiration, but exhalation in this case happens automatically. Now in forced respiration you have more effort produced, sometimes even for exhalation. Exhalation is supposed to be a passive process, but in forced exhalation you have deep rapid breathing, and it happens as when you are doing exercise. Now inhalation and exhalation depends on differences in pressure between the air within the lungs and the environment. Now in order for us to bring in air, we use the respiratory muscles that helps to change lung volumes, because it will expand the thoracic cavity, and it will help to create a greater difference between the lungs and the atmosphere. And this difference is going to be always relative to the atmosphere. Now some of the respiratory muscles will be the diaphragm and also the intercostal muscles. The diaphragm is called the prime mover respiration because it will help to bring 2/3 of the air into our lungs. Whenever you are inhaling, you contract your diaphragm, so it will bring the diaphragm down into the abdominal cavity, and it will enlarge the thoracic cavity, and it will pull the air into our lungs. Whenever you are exhaling, you will relax your diaphragm. Your diaphragm will move up into the thoracic cavity. It will compress the lungs. It will increase the intra-alveolar pressure, and you can exhale the air. The other set of respiratory muscles will be the internal and external intercostal muscles. They are synergistic to the diaphragm, and as their name implies, they are located in between the ribs. Costal, ribs; inner means in between. The internal intercostals are muscles for exhalation, while the external intercostals are muscles for inhalation, and they only bring about 1/3 of the air that we inhale. Along with that, we can have accessory muscles of inhalation like the scalenes that helps to elevate the ribs when we are doing forced respiration. So they are synergistic to the diaphragm. Now during forced inspiration, we can also use the erector spinae muscles. We can use the sternocleidomastoid muscles, pectoralis major, minor. We can use also the serratus anterior, and together they help to expand the thoracic cavity, so you increase the thoracic volume. So the accessory muscles of respiration, they are used in forced respiration, as when you are doing exercise or when you are being chased. Another function of the respiratory muscles is to create the Valsalva maneuver, which is basically taking a deep breath, holding this deep breath as you close the glottis, and then you contract your abdominal muscles so you can increase your abdominal pressure, and that will increases the volume within the abdominal cavity, which will push the organs and the contents out, like during childbirth, urination, defecation, and vomiting. So this is showing you the respiratory muscles. So we have here the diaphragm that will descend, increase the thoracic cavity volume, and then it helps during inhalation. Then during forced respiration, you can use these accessory muscles like the sternocleidomastoid, the scalenes, which help to elevate the upper ribs, and also you can use your external intercostals that elevates ribs 2 to 12 to widen the thoracic cavity. And during forced expiration, your internal intercostals will depress ribs 1 through 11, narrowing the thoracic cavity, and your diaphragm will descend, and your rectus abdominis will help to depress the lower ribs, and then the external abdominal oblique will have the same effect as the rectus abdominis. Now for the neural control of breathing, we don't have autorhythmic pacemaker cells for respiration as we do in the heart. So we have to use these centers within the central nervous system that helps to control the rhythm of respiration. Now we usually have between 15 to 16 breaths per minute, and breathing will depend on the repetitive stimulation of your skeletal muscles from the brain, and it will cease if you have damage to your spinal cord above the neck. Now, you have all this interaction between the respiratory muscles that works coordinated so you can stimulate your central nervous system, and then you can produce an automatic unconscious cycle of breathing that it is controlled by the reticular formation that we have it within the medulla oblongata and also within the pons. And in the medulla oblongata, we have two major centers that controls respiration. We have the ventral respiratory group, or VRG, and the dorsal respiratory group, or DRG. Now, the VRG is the one that controls primarily the respiratory rhythm. Whenever you are at rest, you have eupnea, or quiet breathing. And in this case, your VRG neurons will fire action potentials for about 2 seconds. And during expiration, on rest or eupnea, you'll fire action potential every 3 seconds. So your inspiratory muscles will relax. So you will have 5 seconds for the total cycle for inspiration and expiration. And you will have a respiratory rhythm of 12 breaths per minute. Now in your DRG, you will have neurons that helps to modify the rate and the depth of breathing. So this DRG will be under the influence of external resources. Like if you get scared, you will increase your respiratory rate and the depth just because you are being scared, and you stimulate this DRG. Now within the pons, which is the structure of the brainstem above the medulla oblongata, we have other center that is called the pontine respiratory group, or PRG. This will modify the rhythm that it is sent by the BRG, and also will act on the DRG. So it will adapt your breathing to special circumstances, like when you are asleep, when you are doing exercise, when you're talking, or when you have an emotional response. Now, when you have damage, this is an important point, so this is a key point. When you have a damage to both DRGs on your medulla oblongata, when you have a whiplash accident, for instance, and you damage both DRGs in the medulla oblongata, you will produce apnea or cessation of breathing, and the person will die. Now, within the cervical plexus, in our cervical area, in our neck, we have the formation of the phrenic nerves that innervate the diaphragm. So the spinal nerves C3, C4, and C5 forms the phrenic nerves that innervates the diaphragm. So if you still have a whiplash and you damage your neck, you can damage your phrenic nerve. You can damage C3, C4, and C5. And when you have damage to your phrenic nerves, your diaphragm will not work, but you still have your intercostal muscles. So your intercostal muscles will work, and you have something that is called the costal breathing. So damage to your phrenic nerves will force the person to use costal breathing. So that's the other key point. And this is this diagram or this picture to show you your respiratory control centers, where they are located. So here we have the pons with the PRG, and then in here we have the medulla oblongata with the DRG and the BRG. So these pons with the PRG, it is controlled by upper centers within the cerebral cortex. And also, it is controlled by the limbic system. So you can have emotional response controlling your rate of respiration, or also by high centers within the cerebral cortex. And then your PRG can control this DRG and the BRG. And then in here, within the spinal cord at the cervical area, we have the phrenic nerves. C3, C4, C5 keep the diaphragm alive. That's the mnemotechnic to know which spinal nerves forms the phrenic nerve, C3, C4, and C5. So when you have damage to this phrenic nerve, you start using your intercostal muscles. So you have costal breathing. And when you damage these DRGs in your medulla oblongata bilaterally, the person will suffer from apnea or stop breathing, and the person will die. So some things that can control then these respiratory centers. So the levels of CO2. When the levels of CO2 drop, the pH will rise. So you will have high levels of hydrogen levels. And then you will cause your cerebral arteries to constrict. This will reduce the cerebral perfusion. So you will have less blood supply going into your brain, and then this will cause dizziness and the person will faint. This can be brought under control by having the person to rebreathe when someone is hyperventilating, by putting a paper bag that covers the person's mouth, or allows the person to breathe with its mouth and its nose into the bag, so that the person can have more CO2 and the person will restore the consciousness. So when someone is hyperventilating, it's exhaling so much CO2, and that can cause dizziness and fainting. And you can put a paper bag so you can help the person. Now, your central chemoreceptors, the ones that are found within the brainstem, will detect changes in the pH of the cerebrospinal fluid. And then the pH of the cerebrospinal fluid will be reflecting the levels of CO2 in your blood. And when you regulate your respiration, you maintain a stable pH levels, and then your respiratory center will maintain a stable levels of CO2. Now, your peripheral chemoreceptors will be located, as we mentioned in the circulatory system chapter, within the carotid artery, specifically within the internal carotid, and then you will have these carotid bodies, and then the aortic bodies that you have there in the aortic arch will detect the changes in oxygen and CO2, and also pH levels in the blood. Along with that, we have stretch receptors that are found in the smooth muscles of the bronchi and the bronchioles and the visceral pleura that responds to inflation in the lungs. So we have this reflex that is called the hearing brewer reflex, that it is triggered by excessive inflation. And when you have this excessive inflation, you will form a protective reflex that inhibits the inspiratory neurons from the BRG and the DRG, so you will stop inspiration, and then you won't overinflate your lungs. Also, we have input to the respiratory centers by irritant receptors, like the nerve endings that we have in the epithelial cells of the airways. These irritant receptors, if they detect smoke, dust, pollen, chemical fumes, cold air, excessive mucus, it will trigger the bronchoconstriction of your bronchioles, and then you will have shallower breathing, and then you hold the breath, and then you start coughing to get rid of these irritants. So this is the location of the different receptors. So we have the carotid bodies in here, in the internal carotid. Here we have the aortic bodies, and they help to detect these changes on pH or CO2, and then you can adjust your respiratory rate. Now, the ventilation, it is influenced by changes in pressure, resistance, and airflow. The respiratory airflow has these changes produced by similar principles as the blood flow. So if you have changes in pressure, if you have constriction of your bronchioles, you will have the proportional change of gradient or difference in pressure between the 2 points, distally and proximally to this constriction. And the flow of the air or the fluid is going to be inversely proportional to resistance. So the more resistance you have in the airway, the more constriction that you have, the less airflow that you will have. And that will be kind of when you have a constriction. But in normal respiration, you have a difference in atmospheric pressure relative to intraalveolar pressure or intrapulmonary pressure that will drive respiration. So atmospheric pressure is 760 millimeters of mercury at the sea level, or 1 atmosphere, and it will be lower at the higher elevations. Now, Boyle's law, which is the one that says that at a constant temperature and pressure of a given quantity of gas, you will have this change in volume, that it is inversely proportional to this pressure. So when your lungs have a determined amount of gas and the lung volume increases, you will have an increase in this lung volume that will have a change in the intrapulmonary pressure. So if your intrapulmonary pressure is lower than the atmospheric pressure, your air in the atmospheric pressure, the pressure in the atmosphere is going to be greater than the intrapulmonary pressure, and then the air will come into your lungs, and vice versa. So when you increase the pressure in your lungs and it overcomes the pressure of the atmosphere, you will have exhalation. Now, in physiology, the pressure is measured in centimeters of water, but we use this equivalent that 1 millimeter of mercury is equal to 1.4 centimeters of water. Now, during inhalation, your intrapleural pressure will be slightly negative than the atmospheric pressure. Now, the intrapleural pressure is different from the intrapulmonary pressure, and it is more negative than the atmospheric pressure, the intrapleural pressure. So it's about -5 centimeters of water. Whenever you have this drop in the intrapleural pressure, you will have a recall of the lung tissues, and the tissues of the thoracic cage will have this pulling force in opposite directions that will allow the inspiration to happen. Now, the two pleural layers will tend to cling together due to cohesion of water. But when your ribs swing upward and outward during expiration, the parietal pleura will follow them, and then the visceral pleura will stay together close to the lungs, and it will help in stretching the alveoli within the lungs. So then the entire lung will span along the thoracic cage, and it will increase the volume. And when you have an increase in volume, you will decrease the pulmonary pressure, and then the air will move in. Now, another force that helps to expand the lungs, it is explained by the Shor's law. In the Shor's law, you have a volume of gas that is directly proportional to its absolute temperature. So whenever we have 60 degrees Fahrenheit in the air, you will increase its temperature by 21 degrees Celsius during inspiration, and then the inhaled air is going to be warm to 37 degrees Celsius, the core body temperature of our body, and that will help to expand the volume of the air. So every time that we breathe 500 milliliters of air, just because you have warmer temperature inside your lungs, you will expand this volume 500 milliliters into 536, and then this will help to expand our lungs, and you will have more air coming in, so you will have inflation of our lungs. Now, in quiet breathing, the dimensions of the thoracic cage will increase only a few millimeters in each direction, so in anterior posterior direction and side to side lateral direction, and this will be enough to increase the total volume of the thoracic cavity to 500 milliliters, and thus only 500 milliliters of air will flow into the respiratory tract during quiet respiration. So this is what happens when we are inhaling and exhaling with our thoracic cavity. So when you are inhaling, it is like when you are holding a bucket from its handle, and you move it upward. So in this case, your ribs will swing upward, and then you expand your thoracic cavity. Together with the flattening of the diaphragm, you will have increase in anterior posterior direction that you can see and lateral direction. Now, when you're exhaling, you do the opposite effect. It's like if you put the bucket with its handle downward, so you will decrease your anterior posterior and lateral diameter because you are decreasing these diameters as you are rising your diaphragm. And then when this happens, your intrapulmonary pressure increases and you can exhale. And when you are doing this, when you are expanding your thoracic cavity, you decrease the intrapulmonary pressure so you can inhale because you have less pressure inside the lungs than the atmosphere. And in here, when you are compressing the thoracic cavity in the lungs, you increasing your intrapulmonary pressure, you overcome the atmospheric pressure, and then you exhale. Now, exhalation or expiration, it's a passive process. We have a lot of elastic tissue around the bronchioles, and this is enough to compress the lungs without any use of the muscles. So during exhalation, as you are compressing your bronchioles, you will increase the intrapulmonary pressure, so you decrease your volume of air, and then the air will move from the lungs into the atmosphere. Now, in forced exhalation, you definitely need to use muscles. And the muscles that we use during forced exhalations will be the rectus abdominis and the internal intercostal muscles. So you can rise your intrapulmonary pressure as high as 40 centimeters of water. So this is the other key point. So during exhalation, forced exhalation, you use your rectus abdominis and your internal intercostal muscles. Now, if someone has a rupture of the thoracic wall, like when someone has been stabbed, sometimes you can have opening of the alveoli into the pleural cavity. And this will help into the passage of air into the pleural cavity. So this is called pneumothorax. And during inspiration, you'll have sucking of air through the wound into the pleural cavity, and your potential space will become air-filled instead of fluid-filled. And you will lose your negative pressure that you had, the -5 that you have within the pleural cavity, and then that will prevent the recoiling of the lungs, and your lungs will collapse. And then during atelectasis, you'll have collapsing of part or all of the lungs. And then you will have problems of respiration. And this can result when someone has an airway obstruction or when someone has the formation of tumors within the lungs. Now, alveolar ventilation is the process of gas exchange within the alveoli. Not all the air that it is inhaled goes into the alveoli. Only out of the 500 milliliters that we inhale, only 150 milliliters will fill these alveoli, and then you can have gas exchange. Now, during this process, you'll have parasympathetic system stimulation and also sympathetic system stimulation. But sympathetic system stimulation will increase the dead space but allows great airflow. And then you will have more air coming into your alveoli, and then you will have gas exchange. Now, this gas exchange can be prevented in pulmonary diseases in which you damage your alveoli. So, some diseases in which you damage your alveoli will be emphysema, cancer, etc. So, in alveolar ventilation, if a person inhales 500 milliliters of air, 150 will stay within the anatomical dead space and 350 will reach the alveoli. And in order to calculate the amount of air that it is available for alveolar ventilation or gas exchange, all you need to do is multiply 350 milliliters of air that reaches the alveoli times the rate of respiration. So if the person is breathing 12 times per minute, you multiply that 350 times 12 and it gives you 4.2 liters per minute, or 4,200 milliliters of air available for gas exchange. And you will have then this residual volume left in your lungs that cannot be exhaled with maximum effort. This is called residual volume, which is 1.3 liters of air. In order to measure the efficiency of pulmonary ventilation, you will use a device that is called an spirometer. In the spirometer, someone will breathe air in and out, and then you have a record of this air as it's coming in and out, and then you will have different type of volumes that you can see. One of such volumes is the tidal volume, which is the amount of air that we inhale and exhale in every breathing cycle, which is 500 milliliters. And then after you have your tidal volume, after you have inhaled your 500 milliliters of air, you can inhale some extra air. And this is called the inspiratory reserve volume. And inspiratory reserve volume, it can be up to 3 liters. Now, you can exhale as much air as you can, and this is called the expiratory reserve volume. This is 1.2 liters. And this is one type of spirometer in which the person clips its nose and it inhaling and exhaling through this device. Now in this diagram, you can see the different volumes and capacities in the lungs. So this is the tidal volume. So when you see this line goes up, you are breathing in, and when you see this line goes out, down, you are breathing out. So this is the tidal volume, 500 milliliters each time. After you inhale in your tidal volume in here, and you inhale much stronger, you can obtain your inspiratory reserve volume. And then you can exhale all the way as much as you can, and then you obtain your expiratory reserve volume. Now, the sum of two volumes or more can form capacities. So, in the case of vital capacity, is adding your inspiratory reserve volume, your expiratory reserve volume, and your tidal volume. Your inspiratory capacity will be adding your tidal volume and your inspiratory reserve volume. Your functional residual capacity will be adding your expiratory reserve volume and your residual volume. And then your total lung capacity will be adding all of the volumes or capacities. So we'll be adding your vital capacity and your residual volume. And then it seems like we can hold up to 6 liters of air in the lungs. And I say it seems like because at some point we all have according to our age, our gender, our age, our ethnicity, we all have different lung volumes and lung capacities. So whenever someone is doing this spirometer test, you have to enter the demographic data from this person so that the machine can predict specific volumes or capacities according to the data entered for each person. One thing for sure is that males have greater lung volumes and capacity than females. So what is the residual volume? Is the air remaining in the lungs after a maximum expiration is 1.3. Vital capacity, what it is, the total amount of air that can be inhaled and then exhaled with a maximum effort. And you can see the formula in here. Vital capacity is ERV, expiratory reserve volume, plus the tidal volume or TB, plus the IRV, inspiratory reserve volume. So it can be up to 4.7 liters. And this is important to measure pulmonary health. Inspiratory capacity, what it is, the maximum amount of air that someone can inhale after a normal tidal expiration. It can be 3.5. So we can inhale more air after we inhale normal amount of air during tidal volume. Functional residual capacity is the amount of air someone has in their lungs after a normal tidal expiration. Functional residual capacity is equal to the residual volume plus the ERV. It's equal to 2.5 liters. Total lung capacity is adding all of the volumes and capacity. So the residual volume plus the vital capacity, which is 6 liters maximum. A spirometer is the measuring of the pulmonary function and will help to diagnose problems in the lungs like restrictive and obstructive lung disorders. What is a restrictive disorder? Is when you have a reduced pulmonary compliance or the lack of the volumes of air that can enter into your lungs because you have decreased expansibility of your lungs. So your lungs cannot expand as well. For instance, we have pulmonary fibrosis, in which your respiratory membrane, instead of being made out of squamous epithelium, is made out of collagen fibers, scarring within your alveoli. Pulmonary fibrosis, you can have it as a result of having black lung disease, in which the person has been exposed to contaminants like coal, like asbestos, or other particles, or the person, if he has tuberculosis, the lungs can be damaged and have the person being having pulmonary fibrosis. Obstructive disorders is when you have something that narrows your bronchioles or your airways. Examples would be when someone has a tumor that compresses the bronchioles, or when someone has asthma, chronic bronchitis. And these obstructive disorders can be classified as COPD, or chronic obstructive pulmonary disorder. Emphysema, it combines an element of restrictive and obstructive disorders. Other measurements that you can obtain with the spirometer are forced expiratory volume, or FEV, in which you are measuring the percentage of the vital capacity that can be exhaled at a unit of time. Usually is 1 second. So we have FEV1, which it tends to be between 75 to 85% of the vital capacity that someone can exhale in 1 second. And we can have also a peak flow, which is the maximum speed of expiration. And we can have also the minute respiratory volume, MRV, which is the amount of air that someone can inhale in 1 minute. And in this case, you have to multiply the tidal volume times the respiratory rate. So at rest will be 500 milliliters times 12 breaths per minute. That will be 6,000 milliliters per minute of minute respiration volume. MVV, or maximum voluntary ventilation, will be the MRV, the minute respiratory volume in heavy exercise. It can be as high as 125 to 170 liters per minute, depending on how much the respiratory rate increases and the actual tidal volume. It's similar to the cardiac output, guys. The cardiac output can increase when someone increases the heart rate and also the stroke volume. Okay, so some variations in the respiratory rhythm. Eupnea is relaxed, quiet breathing. Usually, during quiet breathing, we breathe 500 milliliters of air into our lungs at a respiratory rate between 12 to 15 breaths per minute. Apnea is the temporary cessation of breathing. Dyspnea is when someone has difficulty in breathing. The person can be having labored breathing or gasping for air, or having what it is described as shortness of breath. Hyperpnea will be having increased respiratory rate and depth of breathing. Whenever you have a response to exercise, pain, or other conditions. Hyperventilation is having an increased pulmonary ventilation in excess of metabolic demand. Okay, so in this case, hyperventilation is different from hyperpnea. Hypoventilation is having a reduced ventilation that leads to an increase in blood CO2, or hypercapnia. Kussmaul's respiration is having deep, rapid breathing, often induced by acidosis of any kind. Orthopnea is having dyspnea, or difficulty in breathing, when someone is lying down. Respiratory arrest is the permanent cessation of breathing, and tachypnea will be having accelerated respiration. And just to emphasize for alveolar gas exchange, so the alveolar gas exchange happens within the alveoli, and it happens because you have a difference in pressure in the air that it is found in the alveoli and in the gas that it is found within the capillaries. Now, this alveolar gas exchange is influenced by Henry's law, that it says that at the air-water interface for a given temperature, the amount of gas that dissolves in the water is determined by its solubility in water and its partial pressure in the air. So, whenever we are inhaling, we inhale the oxygen because you have a greater partial pressure of oxygen in the alveolar air. And the more oxygen that you have in the alveoli, the greater the amount of oxygen that you will be picking up, because you have a decreased level of oxygen within your arteries that are surrounding the alveolus. And you have a greater amount of CO2 within your alveolar capillaries. That is going to lead for the releasing of CO2 into the air, because in the air you will have less CO2. And then at the alveolus, the blood is set to unload CO2 and load oxygen. Unloading and loading of the CO2, of course, involves the reactions of the erythrocytes that we already talked about. So you have then this oxygen in the alveoli and this CO2 in the capillaries, and your red blood cells will stay a specific amount of time within the alveoli for this gas exchange to happen. So It takes about a quarter of a second, 0.25 seconds, for you to have this diffusion of gases. And at rest, your red blood cells will stay 0.75 seconds, 3 times the amount of the necessary time to having this gas exchange. And whenever you're doing exercise, you have less amount of time for the erythrocytes to stay within the alveoli, but it's 0.3 seconds. That's still enough for gas exchange to happen. And one gas exchange does not influence the exchange of gas of the other. So this is a kind of analogy of what happens with Henry's law and alveolar gas exchange. So at initial state, this is the top for panel for oxygen. At the initial state, whenever your blood reaches the alveoli, you have very little oxygen in your blood. And then in the air, in the alveoli, you have a lot of oxygen. So as your red blood cells stay in the alveoli for 0.25 seconds, you will reach an equilibrium in which the oxygen in the air will diffuse into the alveoli, into the capillaries, into the blood, because you have more oxygen in the alveoli than what you had here in the blood. And similarly happens for CO2. So whenever you have this deoxygenated blood in the capillaries that surrounds the alveoli, this deoxygenated blood, it has a lot of CO2. And your air has very little CO2. But as time is passing by, this a lot of CO2 that you have in your blood will diffuse into the alveolar air because you have greater amount in here than in here. So by diffusion, by gradient, the CO2 will diffuse from more to less. So what is the difference in pressure on the alveoli? So you have a partial pressure of oxygen of 104 in the alveolar air versus the 40 millimeters in the blood. And then for the partial pressure of CO2 will be 46 in the blood arriving in the alveoli versus 40 millimeters in the alveolar air. So just because of that, because you have 104 or more pressure in the alveoli than in the blood, so see the difference is 64 millimeters of mercury, the oxygen will move from the alveoli into the blood, into your capillaries that surrounds the alveoli. And then the capillaries in your alveoli have 46 millimeters of mercury of CO2. And then your alveolar air has 40 millimeters of mercury. So these 6 millimeters of mercury of difference will make the CO2 to diffuse from the blood that surrounds the alveoli into the alveolar air. Now in this principle, it is used to give therapy of oxygen in hyperbaric chambers. So these hyperbaric chambers will have oxygen at a pressure greater than 1 atmosphere of pressure. And just because of that, you will have oxygen diffusing more into the blood. This hyperbaric oxygen therapy can be used to treat gangrene or carbon monoxide poisoning. And at higher altitudes, the partial pressure of all the gases are lower. So you have less diffusion of oxygen into your blood. So the person will be more hypoxic. So this is then the differences of pressure within these gases. So in the expired air, you have a partial pressure of 116 millimeters of mercury, and then 32 millimeters of partial pressure of CO2, and in the expired air will be 159, and for oxygen, and zero point 0.3 millimeters of mercury for CO2. So that's for the expired air and the inspired air. And in the alveolar air, these pressures will change. So 104 for oxygen and 40 for CO2. And then because of this, you will have oxygen loading and CO2 unloading. Because this deoxygenated blood will have a partial pressure of oxygen of 40 and a partial pressure of CO2 of 46. So you will have unloading of CO2 and loading of oxygen. And as you are loading oxygen, your partial pressure of oxygen in the veins is going to change. So you will have now in the arteries 95 millimeters of mercury of partial pressure of oxygen, and you will have a drop in the partial pressure of CO2, so now you will have 40 millimeters of mercury. This will be delivered into your tissues, and then within your tissues, you will have loading of oxygen, unloading of oxygen and loading of CO2. So look at the difference in pressure. In your tissue fluid, you have a partial pressure of oxygen of 40 versus 95 in the arteries. So you will unload oxygen so you can oxygenate your tissues, and then you load the CO2 because you have 46 millimeters of mercury of CO2, and in here you have 40. So there is a 6 millimeters of mercury of difference, and then you can load your CO2, and then you have systemic gas exchange, and then in here you have alveolar gas exchange, and then in here in this part you have gas transport. So in general, CO2 is more soluble than oxygen. That's why you require less difference in partial pressure for the gas exchange to happen with CO2. And the membrane surface area in the alveoli is 70 meters per square meters. So you only have 100 milliliters of blood in the alveolar capillaries that helps to diffuse these gases. But this surface area, 70 meters square meters of alveoli, can get reduced, and then you can have less gas exchange. It can get reduced in diseases like emphysema, because emphysema, what it does is you have destruction of the alveolar walls. Lung cancer similarly, tuberculosis, you have fibrosis of your alveoli, and then you have less gas exchange. So this is what the alveoli normally look like. And in pneumonia, you can have fibrosis and you can have accumulation of fluid, so you have less alveolar membrane to have this gas exchange. And in emphysema, look how many alveolar walls are lost. Not as good as this one, so you will have less gas exchange as well. The alveolar membrane is very, very thin, 0.5 micrometers in thickness, and it will present very little obstacle for diffusion. But if someone has pulmonary edema, like when someone's heart doesn't pump as well, like in left ventricular failure or cardiac heart failure, you will have accumulation of fluid inside the alveoli. So instead of having air, you have fluid, and then you have less gas exchange. And as I mentioned before, in pneumonia, you can have fibrosis or thickening of this respiratory membrane. So instead of having 0.5 micrometers in diameter, of thickness, you can have 1, and then your gas exchange happens less rapidly. So when the membrane is thicker, gases have farther distance to travel, and then it cannot equilibrate fast enough to keep up with blood flow. And lastly, we have also a phenomenon within the blood vessels when you have changes in ventilation. So we have something that we call ventilation perfusion coupling, which is the ability to match airflow to changes in blood flow or vice versa. So when you have gas exchange, you require both good ventilation and good perfusion in your capillaries. And your pulmonary blood vessels can adapt the diameter depending on the airflow to a given area in the lungs. For instance, if any area of your lungs has poor ventilation, so you are not bringing enough air, your pulmonary vessels will constrict because there's not so much oxygen to pick up. And vice versa. If you have good ventilation, your blood vessels will dilate. And then the bronchi will change the diameter depending on the blood flow to an area. For instance, if your bronchi is receiving good perfusion, you have bronchodilation, so you can bring more air. So this is to show you your changes that happens during perfusion and ventilation. So if you have decreased airflow into a specific area of the lung, you will have reduced partial pressure of oxygen in your blood vessels. This will cause vasoconstriction in your pulmonary blood vessels. That will decrease your blood flow. And then you have poor blood flow, and then you will have a response to increased ventilation. So you will increase your airflow, you will increase your partial pressure of oxygen within your vessels because you have more oxygen to pick up, and then that will lead to more vasodilation, to more increased blood flow, and then you will match your blood flow to your airflow. And then your ventilation can be also adjusted to changes in perfusion. Now your blood vessels. So if you have a decreased blood flow, because maybe you have a blood clot that is obstructing one of your branches of your pulmonary arteries or your bronchial arteries, you will have a reduced delivery of CO2 into your alveoli. So you have a reduction of your partial pressure of CO2 that will constrict your bronchioles. So you will have here a constriction of your bronchioles that will decrease your airflow, and that will create a response. So you will have an increased blood flow that is going to elevate the partial pressure in your alveoli, that is going to dilate your bronchioles, that is going to increase your airflow so you can match your blood flow to the airflow. And lastly, just to show you something that I will demonstrate in one of the lab exercises. So in a healthy lung, you don't have so much damage to your, or you don't have damage to your tissue within your lungs. But when you smoke, you will have carcinoma and well, which are tumors within your lungs. So this is one of the consequences of smoking. So I will show you in the lab some of these lungs. This is all for this presentation. So if you have any questions, just email me. And for some of you, have a good day or have a good night. Bye-bye.